Okay. Hello everyone. My name is Xiaocheng Zeng from Tsinghua University, and the title of my report today is Unveil Cis-acting Combinatorial mRNA Motifs by Interpreting Deep Neural Network. mRNA is transcript from a DNA strand carrying the coding information for protein for further transcription and processing into functional proteins. mRNA therapeutics generally involve the in vitro transcription of target genes and their delivery into the human body, encapsulated in liposomes, which have shown significant advantages in treating a variety of diseases. Two major challenges of mRNA therapeutics. mRNA is prone to rapid degradation and weak expression of antigenic proteins, which are closely related to the various components of mRNA. For example, substances such as RNA binding proteins and microRNAs can bind to the UTR, its untranslated regions, and regulate the translation process of RNA. Therefore, studying the binding motifs on mRNA sequences helps us understand which substances will bind to mRNA, which in turn helps us understand the translation process of mRNA. Various biological experiments have been designed to discover RBPs and microRNAs binding motifs. Typically, immunoprecipitation-based technologies are commonly used to identify RBP targets and binding sites across the transcriptome. However, this approach entails high experimental costs, lengthy timelines, and significant technical challenges. Furthermore, these methods are limited to a single motif. Discovering relevant motif combinations still remains challenging. In recent years, deep neural networks have shown successful applications in predicting MRI functions, outperforming traditional methods that are based on feature engineering, indicating that neural networks' ability in deciphering motifs and complex regulatory grammars in mRNA sequences. However, neural networks are black boxes that make it difficult for researchers to understand the rules learned by models. Therefore, we can apply explainable AI to interpret a neural network to get new insights learned by it. We proposed a comprehensive pipeline tailored specifically for the interpretation of RNA sequence to function models. We have also proved that the utilization of our method enables the cost-effective acquisition of crucial regulatory codes in MR translation. Additionally, we carried out simulation experiments. The results reveal that neural network can hardly learn meaningful motif interactions from datasets composed of randomly generated sequences. The method we proposed consists of three steps. The first step is to interpret the relevant motifs from the neural network, which involves identifying the combinations of motifs. The second step is to analyze whether the contribution of each motif is positive or negative. The third step is to construct motif mutagenesis to evaluate the interactions between motifs. Our study utilized two datasets. One focused on mRNA half-life, and the other on mRNA translation efficiency. And the translation efficiency is represented by mean ribosome load, MRL. We applied the same architecture for both half-life and MRL prediction which is a hybrid convolutional and recurrent deep neural network. Here we refer to these two models as a half-life predictor and MRL predictor, respectively. We applied neural motif to discover motifs and motif combinations learned by neural networks. Neural motif is a filter visualization and clustering-based interpretation method, which is specifically designed for deep learning interpretation in the genome field. Overall, the interpretation process can be divided into two steps. The first step is sampling, as can be seen from the left part of the picture, where for a specific neuron or a filter, we need to collect representative samples from the sample space that result in varying activation levels for that neuron. We applied the gradient-guided genetic algorithm to maximize the neuron activation and collect various samples during the optimization process. In the second step, we then perform layer-by-layer clustering on the collected samples. We will send the collected samples into a network and obtain the intermediate feature maps of each layer. We will then start clustering these samples based on their deeper layers feature maps, and gradually proceed to cluster based on shallow layers feature maps. In detail, we won't carry out clustering on every intermediate feature map. However, we only utilize the input feature map of max pooling layer. The reason why we only perform clustering on the max pooling layer lies in the fact that max pooling layer contributes to pattern merging. With the neural motif, we can obtain the motif preferences for each neuron. That is, which motifs can activate a certain neuron. Shallow layer neurons typically recognize individual motifs, while deeper layer neurons often identify combinations of these motifs. After that, to ascertain which query motif found by neural motif exhibit significant similarity to allotted RBP binding motifs or microRNA binding motifs, a motif similarity was conducted with TomTom. Here, we only present the interpretation results of half-life predictor. We compare the neural motif with other interpretation methods from two perspectives: motif diversity and motif quality. As for motif diversity, we collected the motifs found by each interpretation method and applied a Venn diagram to illustrate the motif diversity of these methods. As for motif quality, we measured the quality of the query motif by assessing its similarity to the database motif. The lower the Q value, the more similar the current query motif is to motifs in the database, indicating higher quality. Through interpreting the MRL predictor and half-life predictor, we identified motifs and motif combinations that align with prior research and established biological prior. For example, here has been proved to bind to 5' UTR. AOH elements in the 3' UTR are the most common determinants of RNA stability in mammalian cells. RMB4 element is known to suppress a cap-dependent initiation process. It implies that our interpretation method is effective in interpreting crucial biological motifs in mRNA. Although we have obtained motifs learned by CNN, it is necessary to determine whether these motifs have a positive or negative contribution on mRNA stability or translation efficiency. Additionally, the effects of individual motifs will contribute to our subsequent analysis of motif interactions. For specific neuron X, it's a thin filter. We divide the dataset into two groups based on whether the samples can activate neuron X or not. Then we conduct a statistical significance test to compare the means of the mRNA properties from the two groups of samples. If the mean of the group that activates the neuron is significantly higher than the other group, it indicates that the visualized motif for IAG from this neuron X has an enhancing effect on mRNA properties like half-life or MRL. Otherwise, this neuron X has an inhibitory effect. Neural motif has efficiently screened the most potential motif combinations for us. Then we utilized a combinatorial in silico motif mutagenesis approach to assess epistatic interactions between pairs of candidate motif instances. Here, we denoted as motif A and motif B. Motif A for the GGAC and motif B for the UGUSA. Within a sequence, we represent model predictions as lowercase o for intact sequence containing both motif A and motif B. Lowercase b for motif A scrambled, and lowercase a for motif B scrambled, and lowercase n for the Lao sequence, both motif A and motif B scrambled. Then we conduct a Wilson signed rec test on the paired values for all instances of a motif pair and obtain the interaction type. We primarily consider three types of joint effect, namely additive, synergistic, and antagonistic interactions. Additive interaction represents that the contribution of two motifs when they exist alone sums up to the contribution when they exist together. Synergistic interaction represents that the combined contribution of two motifs when they exist together is greater than the sum of their individual contributions when they exist alone. Biological antagonistic interaction represents that the combined contribution of two motifs when they exist together is lower than the sum of the individual contributions when they exist alone. We used an in silico motif mutagenesis analysis method to quantify the influence of pairs of co-occurring motifs on MR half-life, and we generated an interaction map to illustrate the interactions between motifs. As can be seen from the left figure, in the interaction map, the blue dots represent that the motif A and motif B are independent from each other, indicating low interaction, and green and orange dots represent motif A and motif B interact with each other. Other blank space represent motif pairs for which the combinations was not found by neural motif. And besides, the right figure means that the M6A motif and the PUF element had interaction with each other. As can be seen from the left figure, which is the interpretation results of MRL predictor, we found that the UAUG is a shortcut for a model to make predictions. Since the interpretation method results primarily display repeated UAUG as the most common motifs. The right figure shows that the probability distribution curve of the MRL on the original dataset and the AUG-free dataset. The left peak of the blue line corresponds to sequences containing AUG motifs. As expected, the left peak disappeared in the distribution curve of the mRNA on the UAUG-free dataset, which suggests that the AUG is a significant factor influencing mRNA. Therefore, UAUG is the dataset bias that we should remove a UAUG before training. Our mRNA predictor is based on randomly generated 5' UTR. However, randomly generated 5' UTR sequences rarely contain strong motif combinations. Here we perform a simulation experiment to validate this hypothesis. We generated two datasets, one with random sequences and the other with manually designed sequences. A random sequence means that each nucleotide in the sequence is randomly selected from HCU. The design sequence means that the sequence always contains a pair of motifs. Subsequently, we trained and interpreted two 3-layer CNGRU on each dataset. As expected, the model based on design dataset learned all 6 interactions with a true positive rate of 100%, while the model trained on the random dataset didn't discover any interactions. In summary, our pipeline can provide reliable insights into the motifs and their interactions that neural networks have learned from biological data. We find that the randomly generated sequences are less informative for discovery of motifs and motif interactions. And there is still rich information learned by the model and can be further explored, such as motif indication order and number. Finally, you can replicate our work through the code and data provided in the GitHub link. Finally, I would like to thank my co-author Wei Zheng for his contributions to this work and my Professor Wang for his guidance. I would also like to thank the lab mates from Wang Lab. And this work was supported by the Beijing Municipal Natural Science Foundation and the National Natural Science Foundation of China and the grants from the Guoqiang Institute in Tsinghua University. Thank you for your listening. That's all.